Lauc. He's a professor of biochemistry and molecular biology at the University of Zagreb. He is also the director of the National Center of Scientific Excellence in Personalised Healthcare, honorary professor of the University of Edinburgh, the King's College London, and member of the Johns Hopkins Society of Scholars. In 2017, he initiated the launch of the Human Glycoma Project and is one of the two co-directors. His topic for today's conference will be big data in personalized medicine. Since I already see him here next to me, without any other further ado, we will give you the stage, but just a short video before.
Thank you.
Welcome.
Thank you, and thank you for staying so late to listen to me. I'm not a data scientist, I'm a biologist. But is the echo too strong? Or is everything okay with the sound? It's okay, good. But I will tell you something about from the side of the data user. We use the data which data scientists produce. I do have data scientists in my team, but personally, for me, that's just a black box. So, one of the problems we have in medicine is that 80% of all the smart drugs, but also all the lifestyle interventions, are actually wasted because they are given to the wrong people. And they are given to the wrong people because we do not have good biomarkers which can help us to stratify people into groups who would respond to a specific therapy or even a specific lifestyle intervention. And there is a huge effort in trying to develop these biomarkers, but still, we as a planet are still not sufficiently good at that. And one of the problems is that the majority of people actually focus on genes. So probably over 90% of the biomarker discovery efforts were by sequencing genetic information. We sequenced tens, probably hundreds of millions of people, and still there is a very little translation. So technically, the Human Genome Project was a success. Now you can sequence all 6 billion base pairs for below $100, but we do not know what are we actually seeing. We cannot use this data. And one of the reasons for that is that genes actually do not encode that much information as we believed. Approximately only 20% of longevity and 30% of most complex diseases is genetic. Everything else is something else. And one of the best examples for that are identical twins. So these two pairs of people have every single letter in their genome identical. So every letter is identical. They're identical twins. But their life trajectories are very different. And this is mostly because of a lifestyle. So genes are not enough to stratify people into actionable groups. And one of the big elements which are missing if we are looking only at genes are the glycans. And glycans are the field where I devote most of my time to. And glycans are actually responsible for the large part of inter-individualization. So glycans are one of the key reasons why we are different. And actually even the first biomarkers in personalized medicine are glycans. So the blood groups, A, B, and O, blood groups are chemically 3 different glycan structures. So we need to look into different aspects of biology, not only genes, to understand what is going on, but we have a big problem. And the big problem has been identified nearly 20 years ago by Diogenes, one of the greatest minds of medicine, who published a very convincing paper showing that nearly all published results from small studies are false. Over 90% of things which is in literature is false. So our knowledge is based on false data because people were doing small studies. I will not go into reasons why this happened, but it's very evident that all the discoveries made in the small studies, once replicated, get dissipated, meaning they got lost. So this was just a random finding. And I had my personal experience in that, and this is when we did this study. We were comparing kids with allergies with healthy kids and got something which is unbelievably different. We nearly published it, but we said, okay, this looks too good, let's replicate it before we publish it. And we never managed to replicate it. This was just some kind of strange whatever happened. We got this completely false information. Unfortunately, many people publish this type of data, and this is why the scientific literature is plagued with the false data. So if we want to do good science, we have to move from small studies to large studies. And this is not easy, especially not for biologists, because we have to learn all the strange terms which we never used before, like normalization, batch correction, structure of error, randomization, blocking, and so on. So we had to work together with statisticians and data scientists to develop these methods. And personally, for my lab, this was 10 years, 10 million euro endeavor to come where we are now. In the field of genetics, this revolution started slightly over 15 years ago with the so-called genome-wide association studies. So when instead of asking the question, is this gene relevant for a specific disease, we started to ask questions, which genes are relevant for a given disease, and generate huge amount of data, which we then analyze to try to understand the biology. But this brings another problem, and this is when you use data science, when you use modeling, when you use any kind of algorithms on the data which you cannot easily check by kind of a common sense, then all this modeling can be very, very wrong. And this is something we painfully learned in this pandemic. So this is, for example, official UK modeling of the number of hospitalized people in the last wave. And this was the central prediction. They claimed this 95% certainty. This was kind of catastrophic, and this was kind of over-optimistic. Well, this is what actually happened. So, complete nonsense was generated by analysis of huge amount of data, because there were no ways to use a common sense to check what is coming out. I'm not saying that all the analysis is bad. Of course, there are some examples of fantastic data science which helped in this pandemic, like the Zoe study led by the Tim Spector, who practically showed that by collecting symptom data, just people logging how do they feel, you can get equally well monitoring of the dynamics of the pandemics. You can get much faster information about the change in this R coefficient completely for free. So instead of hundreds of billions of pounds, which was paid by for PCR tests, this gave exactly the same information practically for free. What we did in field on COVID was that in spring 2020, we knew that the majority of respiratory viruses are highly seasonal, but nobody knew for SARS-CoV-2 virus. And it was too dangerous to make public health decisions or governmental decisions based on assumptions. So we went to all our colleagues in the hospitals, collected data about individual patients, because at that time, testing was very unreliable. It was impossible to compare testing between different countries. So we went for patients who were already infected and checked what is going on as the spring came. And in Barcelona, it was unbelievable. So there was a drastic decrease in the length of the stay in a hospital. And of course, we cannot make conclusions from one example. Maybe this was just the wrong, something wrong with this data. But then we replicated it in Bergamo, in Nottingham, in Coburg, in Zagreb. We noticed that as spring is coming, people have much shorter and much less severe symptoms of COVID. And this was one of the evidence which enabled Croatia to open very quickly in 2020, also in 2021, which also enabled us to now behave normally because our governments trust our data. But the idea is, Big data plus sanity check. And sanity check here was the basic biology. This is your mucosal barrier in your nose. When it's fully hydrated, this is removing all the microbes from your airways. And here is what happens when you dry it out. This is what we do in winter. In winter, we dry our mucosas because we don't think about air humidity, and this is why the bugs stay in our airways and make us ill. Another example, 30 years earlier, when we learned the same thing, and many of you are probably not too young to remember that, we had a huge problem in the late 90s with a very large number of victims in a mass graves which were unidentified. And then we had 3 DNA laboratories. I was leading one, Dragan Primorac another, and Milovan Kubat, one in Zagreb. And we actually had the most successful program of identification of victims from the mass graves in the world. Because what we did, we used a sanity check. We did use the DNA data, but in addition to the DNA data, we said, okay, is this data logical? So is this body found on a location where the person was killed? If somebody was killed in Dubrovnik, you don't expect to find the body in Vukovar. Or is this person 1 meter 80 or 1 meter 60 tall? Just this basic information plus DNA evidence enabled us to be way more successful than, for example, International Commission for Missing Persons in Bosnia, who were just using the DNA data. So in the second part of my talk, I will talk about nicer things. And this is healthy aging. And how can we live longer and healthier, and how can we use data in this field? And this figure is actually very deceiving, because when we look at the average life expectancy, we think that humans are living longer. This is actually not true. The average human is living longer. But even in the old ages, some individuals were living up to their late 80s or even 90s. So human biology did not change in the last couple of hundreds of years. It just, the environmental conditions in which we live change for the majority of people. So now even the normal, the common people can have the privilege of the noble people from the ancient time and live long and healthy life. And it is actually our decisions and our lifestyle which is deciding when are we going to get ill and for how long we are going to stay healthy. And here it's very important to have the objective measure to evaluate what is actually going on. And slightly less than 10 years ago, on December 10th, 2013, 2 biological clocks of aging were published. One was a epigenetic clock, published by Steve Horvath, and the second was a Glycan clock, published by Group. Incidentally, on the very same day they were published. An epigenetic clock is typical data-driven clock. They measured 300,000 individual data points and model it based on chronological age. And actually they were very accurate in modeling chronological age, and today there are numerous tests which are commercially available. The only problem is they actually do not disclose what are they measuring. So out of these 300,000 points, each of them is measuring something different, so you cannot mix and match. So if you take one, stick to that one test, you cannot mix it. Our Glycan clock of aging, and I have to disclose here, of course, I'm highly conflicted here. I'm inventor and a patent, I'm co-founder of a company. This is something that I'm actually making money out of it, so you don't have to buy it because I say it's good. And the test is based on Glycans, modifications of immunoglobulins, which either make immunoglobulins anti or pro-inflammatory depending on the situation. And interestingly, these 2 clocks highly correlate because they correlate with age, but acceleration, epigenetic aging, and Glycan aging does not correlate. So these are kind of 2 different aspects of aging. So to simplify it, gene is a blueprint. So gene is an information telling how the protein can be built, like this Batmobile. This is the blueprint for a Batmobile. If we make it only of a protein, so the polypeptide part, you get this old Vivo car. It moves, but not really a good car. If you add all the modifications to it, including the glycans, then you turn it out in a hybrid car. So gene, protein, glycoprotein. And then there is epigenetics. Epigenetics is an instruction manual. This is telling our cells what to do with each of the genes. So kind of a booklet telling you how to use the car. Glycans that I work on are these hairy modifications of a protein. So this is the famous S-glycoprotein, which stopped the planet for the 2 years. Unfortunately, many scientists look only at the polypeptide part. This is even the director of NIH at that time tweeting about the S-glycoprotein without a single glycan. Well, actually, glycans are more surface of the glycoprotein than the polypeptide. So I usually use this analogy. So yes, you can do a lot of good research on this bird, but however well you plan and execute your study, you will never see this bird flying, because bird without feathers cannot fly. So a lot of science can be wasted here without even understanding that flight can exist, because people ignore glycans. So glycans are essential to understanding many things. Problem with glycans, very difficult to analyze. You need many chemical steps. Not so many labs do it. Actually, my lab did the first study 15 years ago, and we are currently leading this global effort. We analyzed over 150,000 different people from some of the best phenotype and genotype cohorts, and one of the first things we learned is, of course, glycans associate strongly with age, but even more importantly, the difference between the glycan age and the chronological age associates with the biomarkers of unhealthy life. So people who live unhealthy life age faster in their glycan, IgG glycosylation. People who live healthier life age slowly. And we know from numerous studies that adding additional healthy habits, this is 0, 1, 2, 3, 4 healthy lifestyle habits, can add up to 10 years of healthy life. So by changing your lifestyle even in the 50s, living healthier, you add yourself 10 years of healthy life. And the emphasis here is healthy. And also, if you do it for a longer time, it would be even more. This is a famous example of two monkeys. This one is on a caloric restriction, so he's hungry for the last 2 decades. This guy was happy, he was eating, but he aged much faster. I will not go into discussion what's better. Personally, I will not be hungry for 20 years to live a little bit longer, but some people decide to live longer even they're hungry. But not everybody is so much food dependent. In some people, aging is driven by stress, or by the absence of sleep, or the absence of the physical activity. So we are all different. There is no standard human, and we have to find whether we are the diet guys, so we have to be hungry, or maybe we should just sleep more or be under a little bit less stress. And these same glycans which drive aging, they also drive many different diseases. So we studied, as I mentioned, 150,000 people. Many of them were case control cohorts. So these last 4 bars, this is the effect of aging. This is what happens to your glycans as you age. These other bars are what happen if you get arthritis, IBD, diabetes, colorectal cancer, and so on. So people with the disease look older than the healthy people of the same chronological age. And we published many papers in that field, I think over 200, and we really tried to publish some of them in the leading journals, including the Circulation, Gastroenterology, some of the Nature journals. One example, cardiovascular diseases. So our first large study was published, I think, 4 years ago. We studied 3,500 people, and we have shown that IgG glycan composition correlates with the cardiovascular disease risk score, so the risk to get cardiovascular diseases. We got samples collected 30 years ago with a clinical follow-up, and actually could predict better than any other biomarker who will and who will not develop either heart attack or stroke. And in some animal experiments, just fixing the glycans with one dietary supplement, unfortunately not available for humans yet, you can actually prevent hypertension in obese mice. So they can eat and still not get hypertension. The similar thing is for diabetes. Glycans can predict diabetes to a significant extent, and these things happen gradually. So this is 10 years before before diagnosis, 7 for at diagnosis. So glycans change as you are approaching the disease phenotype, and kind of general model is disease is an endpoint. Disease is a name which you get when you go to a hospital saying, you know, I have pain here, they do some studies, and then you get the name of your disease. This is beyond the point of no return, most of the times. Once you get a chronic disease, you will have it till you die. But disease develops through years or even decades. And often we see changes in glycans 10 years ago, 10 years earlier before disease is developed. So here, a lifestyle intervention on a simple preventive intervention can change this direction, and you never go here or you go there much later, and you stay healthy for longer. And one of the important elements here is inflammation. Inflammation is one of the main drivers of complex diseases which are burdening the modern society. So the question here, can we revert it? Is there something we can do to change this direction and to age slowlier or to develop diseases later? And as I mentioned, Glycan Age is a commercial product. There are hundreds of clinics selling it globally, and this is one of the anti-aging clinics in New York, Joe Raffaelli, and most of his patients, after he gets them in the treatment, end up 20, 30 years younger within a year in the hospital. This is Tim Spector, one of the famous genetic epidemiologists, the one who actually developed the Zoe application for COVID I was mentioning before. He was reasonably good, a little bit too stressed here, decided for intervention, lost 11 Glycan years in 6 months. If you want to know what he did, there is an Instagram live which we did together where he was talking about his intervention. Weight loss is an efficient way. Bariatric surgery is a drastic approach. They make your stomach smaller so you cannot eat a lot. But one of our patients lost 37 Glycan years within 6 months. Most of them get better. Okay, we don't want to get surgery, but also the regular weight loss helps. So this was 2,000 twins tracked through 15 years in 3 time points. Those who were gaining weight were aging faster. Those who were losing weight were aging slowlier. Caloric restriction, this poor hungry monkey definitely works. So if we eat less, we will live longer and be healthier. That's a sad fact. But when we talk about the different diets, there is no magic diet. So there is no single diet which works for everybody. This was 1,000 people on 5 different diets. And on each of the diets, some got better, some got worse, and you could not tell up front. So we are different, and we have to find for ourselves what works. The same goes with for exercise. We have been taught that you can sweat out the extra food. This is false. You cannot sweat out the bad diet. Diet is much stronger effector than exercise. Exercise is important, but too much exercise is actually doing damage, and we are seeing this in a number of studies. Gut microbiome is extremely important. We are living together with our microbes for millions of years. Nobody can survive with the microbes, so we have to think about the microbes. We did interesting study of fecal transplantations. You take feces from one person, put it in the guts of another person. It can actually improve your glycome. And as my final slide, the whole idea behind what we are doing is that we are developing information, sources of information, which integrate genetic, epigenetic, environmental factors into something which can be quantified, which can be used by a person in a way they can understand. Meaning, this is younger, this is older, this is good, this is bad. You measure it and check what is going on. And we burn a huge amount of money. We spend between 2 and 3 million euros testing different people every year. This was funded by a number of different research grants, which I have to acknowledge here for their generous support. And I thank you all for your attention.
Thank you, Professor. Are there maybe any questions? No, really? Oh, there are 2. Great, 3. Can we please get a mic?
Is it
Try
working?
it.
Yeah, I had a question regarding the preventive potential of the glycan. What you said, for example, if someone was to have a chronic disease, but it can be seen through the glycan age 10 years before. So, for example, this was me, and 10 years before I had a high biological glycan age. Is it possible from the data to see on the glycan exactly what is wrong with me, or is it just a general score that says, okay, you need to check out?
Yeah. Unfortunately, we cannot say what is the source of the bad IgG glycosylation. We can say it's an inflammatory process. We don't know where is it coming from. There can be many different sources. But what we can say is after you try something, whether it works for you or not. For example, metformin works in some people. Go on small doses of metformin, it might help. Hormone optimization works in other people. Weight loss works in some people, not everybody. There are skinny people who don't look good. There are some obese people who look good. So it's at the moment, the only thing we can do is tell you if what you are doing is helping or not.
Sorry, just one short follow-up question. How quick is the change reflected in my glycan?
Depending on what you do, if it's very aggressive thing like a hormone intervention or a drug, it could be 6 to 8 weeks. For the normal things like exercise, weight loss, it's usually 3 to 4 months.
Okay, thank you.
We have another one.
Hello, one question here, slightly unrelated to your presentation, but still from the same field. 4 or 3 years ago, AlphaFold was produced for predicting this protein structure. Did it turn out to be useful for, let's say, academia and so on? Or?
What does it mean, useful? So definitely, predicting protein structure is still a big challenge. But these things are moving forward, and of course, different algorithms are helping it. But one of the big problems there is that the majority of these predictions are actually ignoring glycans. You just cut off the glycans and you try to predict. And this is one of the big obstacles at the moment.
Because as far as I know, it takes a lot of time to check this protein structure by hand.
Yeah.
Do you see all the future in this direction here?
Definitely. There is a huge future in any of similar approaches. The only problem is that once this all becomes a black box algorithm, which cannot be what I said, sanity check. Now, is this something which looks reasonable or not? And I think we are approaching the situation where in the entire medical field, decisions will be made by different algorithms, which can make things very wrong. So they can be fantastic, but the moment we lose kind of way to check whether something is okay or not, then we end up with those predictions I was showing. And it's, I don't know how will this be solved. But eventually it will. There will be maybe 2 different approaches, and then if they match, you say it's okay.
Okay, thank you.
I think there's one question in the back.
Hi, so I actually had the same question, but I'm going to try to come up with something new. You mentioned that there was a study where a professor found most of the research was basically flawed, and did I get it correctly? Right? So the question is the flaw maybe because of the underlying complexity of the system?
No, there are many reasons. One of the biggest reasons is the way we get funded and promoted. So when you're doing hypothesis-driven research, so I'm a big professor, I have a great idea, and then I tell my great idea to my PhD student to work on it for 3 years. If I'm right, the guy will get a PhD, he'll get a great paper, get another great job, I'll get a new grant, I'll get promoted. If I'm wrong, we're all in trouble. So I'm trying hard that he confirms my hypothesis. He's trying hard to confirm my hypothesis. So there's always a little bit of cheating. Like, at this point I made a mistake, I'll cross it out. The second thing is multiple testing. So So if all of us will be flipping coins, somebody will get 5 heads in a row. And maybe that person will decide, oh, this is a magic coin. I will now have a coin which always gives a head. And if he does not try it again, then this goes into literature and then there is a magic coin. And unfortunately, this was happening for decades. You do a difficult experiment, 100 people do it, 99 of them don't see a link, they don't publish. The one incidentally sees something and publish. And then nobody repeats it because it's done already. And this created this huge burden of false data. Only a few years ago, there was a consensus that you cannot publish anything until it's really replicated, or you cannot publish in a good journal. You can publish anything in a junk journal. There are 250,000 studies on COVID in the last 2 years. That's crazy. And this goes for everything else.
Okay, thank you.
Are there maybe any more? One more? I see you.
Hi, sorry, I know you said repeatedly that the data science part is a black box to you, but I think everybody here would love to hear anything more about that, like what's the role of the black box, what do you use it for, anything, just to clarify it a little bit.
So it's so all the methods are black box for me because I don't know how to code. I can't write anything. I was doing it 30 years ago, but I can't do it anymore. So what we are trying, what my data team is using, all the tools there are, is just to try to understand patterns in the data. So we measure a huge amount of data, and then we try to distinguish signal from noise, because there's always noise, and then as the measurement is getting better, there's less noise and more signal, but at the beginning you see a lot of noise, and then you try to understand what is the underlying biology. So more or less this is what we do. So we measure a huge amount of data, and then try to see a biological signal which is causing some kind of signal in the data, and then we try to understand it. So I say it's a black box because I talk to my data people and say, okay, do some magic. Take these 200,000 data points and show me something which I can check with my eyes and see whether there is information. So whether there's the best way, I'm not sure, but I'm a little bit too old to learn new tricks.
We had one more at the back over there. Just please raise your hand once again so our volunteer can see you.
Thank you for your presentation. I have one question about Glycans. I'm wondering, is there a big data approach on Glycans, and so far how successful it is, and is there, if it is that also one direction in the future?
Sorry, I missed the second word. Is there a...
Is this a direction, if that is correct, that this big data is approach on the Glycans research?
So, depending how you define a big data, so we don't generate that much data. Some people, what are they trying now? So usually what we do, we measure something, and then we do collect thousands and thousands of data points, and then we try to distill it into what we understand, and then analyze. Now, I just recently got an idea from, actually a proposal for collaboration from one group in US, who want to take all the data that we measured and then use different ways to mine this data. So not use the first biological knowledge to distill the data, but take the entire data set and try to find something. If I got your question right, definitely the Glycan field is still too small compared to genetics. Actually, out of 150,000 analysis in a recent review, my lab generated 120,000. So it's still largely driven by us, but it is moving forward in that direction.
And can I ask one more question? Is that possible? So you said big data processing can be looked as a black box and can be very wrong. So my point of view, there can be some process of validation of data. So you make a process on big data, then you sampling random samples, smaller random samples, and compares to that result you get from the big data.
Yeah, so generally this is what people do, but the problem is that majority of algorithms which exist were written often for something else. And then there are assumptions which may not be met in the new data set. And the key problem is that people apply this, I call it black box, or the algorithm or approach, without really understanding what are the constraints in the data. And then you can get something completely wrong. So, of course, yes, you do all these things like, I don't know, cross-validation or whatever approach to verify, but the problem is if you have used the wrong tool. For example, people now mostly do analysis in R. And there are different R packages which are finished, you know, everything is written, just take the package and apply it. And of course, in some kind of description, it will be told, you know, I don't know, this cannot use if the data is constrained in some way. But people don't check it and use it on the wrong type of data with the false assumptions, and then things go really wrong. And as the tools are getting more and more complicated, it will be more and more difficult to understand whether you're applying it on the right set of data or wrong set of data. And as a biologist, I really have problems understanding the different, for example, normalization techniques. I can't even remember the name. There's something like quantum normalization, this type of normalization. That's too much statistics for me. And on the other hand, the statisticians don't understand the biological questions. So data scientists and biologists have to work together to kind of integrate our knowledge.
Thank you for your answers.
Are there any more? No. Thank you, Professor, very much. This is for you.